Hello guys and welcome back to Excelsior Academy. So in the previous lecture, we have discussed about like what are the release factors and how the termination of translation will occur, right? So proceeding with that only, we are going to discuss more in detail about the termination, like how exactly these release factors like class 1 release factors, class 2 release factors are helping in carrying out the termination. Okay. Yeah, so let's talk about first class 1 release factor. So if you remember, class 1 release factors can identify few codons and then it can stop the translation, right? So how it is happening? Because these class 1 factors, they share a 3 amino acid sequence, that is GGQ. If you remember, in the previous lecture we have discussed like where we had got the tRNA and this release factors, right? And what we saw at the bottom, we have got the anticodon peptide, which can base pair with the or which can recognize the stop codons, right? And on the top, we had this GGQ motif, that is your glycine glutamine. Now, this is important. This motif is important because it is required for the release of the polypeptide. Now, ये polypeptide कहाँ से release होगा? From the tRNA which is present at the P site. Got the point? Because now at the A site, it is empty. There is no tRNA. Why? Because stop codon has come, and we know that there is no stop codon for your there is no tRNA for the stop codons. Okay. Now, the structure of release factor 1 bound to the ribosome confirms that this GGQ motif is located in close proximity of the peptidyl transferase. So if you remember in the structure, we have seen like how nicely this GGQ is present at the tRNA which is present at the P side. So if you remember the CCA, that is the acceptor arm of your P side tRNA, okay, and the GGQ motif of your release factor 1. So they are present in the close proximity. Okay, so this will help in the release of the polypeptide chain. Now, how it is going to happen? With the help of the hydrolysis. Okay, now class 1 release factors functionally mimic a tRNA. So अगर हम structure देखेंगे जैसे हमने पिछले class में देखा था, the structure of the class 1 release factor is almost like a tRNA. So it have got a peptide anticodon which is going to interact with the stop codon and the GGQ motif which reaches the peptidyl transferase center. So I hope this much is clear, Tanvi. This point is really very important with respect to your exam point of view. Is this point is clear?
Yes, ma'am.
So in the previous lecture also we have seen how the structure mimics the entire structure of the tRNA. So this is just a repetition of the same structure. Now, sorry, what happened? Okay. So here if you see in the structure, you can see that ये जो wine color में है, this is your tRNA. Okay, this is your tRNA. And what do we see? The other one is your release factor 1. ठीक है? Only this portion is थोड़ा सा extra. Otherwise, if you see, it is having almost the structure similar to your tRNA. तो यहाँ पे आपका peptide anticodon हो जाएगा, and यहाँ पे आपका this one is having the CCA and the GGQ motif आ जाएगा. Okay. Now, let's talk about the class 2 release factor. Okay. So once a class 1 release factor has triggered the hydrolysis of the polypeptide chain or the peptidyl tRNA linkage, it must be removed from the ribosome. ठीक है? क्या remove होना चाहिए? Class 1 release factor का क्या काम था? Polypeptide chain release करना from the ribosome mRNA tRNA complex. Correct? Okay. तो पहला काम क्या है? Class 1 release factor का to remove or release the polypeptide chain. Second क्या है? The class 2 release factor, that is RF3, which comes and helps in the release of the class 1 release factor. Are you able to get the point? ठीक है? RF3 क्या करता है? It helps in removing the RF1 from the ribosome tRNA mRNA complex. Okay. Now this is done by the RF3. So RF3 is a GTP binding protein, but if you see, हमने जब last time discuss किया था, so the elongation factor, if you remember, the elongation factors, what are those elongation factors? So we had two elongation factors, if you remember. It is called EF2 करेक्ट। एंड दूसरा क्या था आपका EFG, राइट। इसमें हमने क्या डिस्कस किया था, दैट ऑल these two elongation factor they are bound to GTP, इनकी एक्टिविटी के लिए दे नीड GTP, ठीक है। सिमिलरली RF3 also needs GTP binding, it is also GTP binding protein, but it has got higher affinity for GDP as compared to GTP. elongation factor के केस में क्या था, EF2 is bound to the GTP, then it has got the high affinity. The moment hydrolysis happens, okay, that what will happen, it loses its affinity and gets released from the complex. Just उसका उल्टा, ठीक है। Thus, free RF3 is predominantly in the GDP bound form, and RF3 GDP it binds to the ribosome in a manner that depends on the presence of the class 1 release factor. इसका मतलब क्या, जब आपका RF1 factor is bound to the ribosome mRNA tRNA complex, then only the RF3 will come. Okay, and in the cytoplasm it is found in the form of GTP bound. Okay, now this slide is also very important, ठीक है। The same thing we are going to see in the form of the figure also, तो थोड़ा ज्यादा clarity आएगा। Okay, so let's first discuss all the important points and then we can see how the figure is explaining. So after the class 1 release factor stimulates polypeptide release, there is a conformational change in the ribosome and also in the class 1 release factor. This is going to activate or stimulate your RF3, ठीक है, to exchange its bound GDP to for a GTP. Okay, ये first step हो गया. Now these factors exchange GTP exchange factor for RF3 in the same way how EFTS does for EF2. ये हमने elongation factor में discuss किया था, like how GTP exchange is happening for EF2 and EFG, ठीक है। Now the binding of GTP to RF3 leads to the formation of high affinity interaction with the ribosome. Okay, so what does it mean? जैसे ही RF3 में GTP आएगा, TP आएगा, okay, GTP आएगा. So now this has got the affinity for your ribosome, which will ensure that RF1 जो है release हो जाए, ठीक है। Now this change in conformation displaces the class 1 factor from the ribosome. Okay, now जैसे ही ये होगा, it will allow the changes or allow the RF3 to bind to the factor binding center of the last subunit. So if you remember in the ribosome when we were discussing, so you have got the 30S subunit, आपका mRNA है, P में आपका E साइड है, P साइड है, A साइड है, और A साइड के पास है आपका फैक्टर बाइंडिंग सेंटर, ठीक है, जहां पे आपका EF2 बाइंड होता है, प्रॉपर्ली tRNA अरेंज होता है, एकोमोडेट होता है, right? So in that site again the RF3 will go and bind. Now as with other GTP binding proteins are involved in translation, this interaction, जैसे ही आपका RF3 goes and binds to the factor binding center, there is hydrolysis of GTP happening, ठीक है। Now this has got less affinity, so in the absence of the bound class 1 factor, the resulting RF3 GDP has low affinity, so it will get released from the ribosome. So eventually what had happened, आपका ट्रांसलेशन स्टॉप हो गया है, पॉलीपेप्टाइड चेन रिलीज हो गया है RF1 की हेल्प से। RF1 को रिलीज किया किसने, RF3 से। So RF3 does not have got good affinity when it is bound to GDP for the ribosomes, right? So again it will automatically get dissociated from the ribosome, ठीक है। Tanvi, are you able to follow this slide? Okay,
Yes, ma'am.
okay, now let's see what we have discussed in the diagrammatic way. Okay, so first thing what is happening over here, so first is the recognition of the stop codon by the release factor, okay. So this release factor is coming and binding over here, you can see that it has got the GGQ motif. So this GGQ motif will ensure the hydrolysis of the peptide bond, polypeptide chain. So this polypeptide chain is getting released, correct, itna clear hai? But right now your RF1 is bound to your A site of your ribosome, correct? Next, what is going to happen? RF3 GDP is going to come. Okay, तो ये आपका polypeptide chain release हो गया, तब तक आपका RF3 that is class 2 release factor is coming and binding over here. Now this will initiate the release of your RF1, ठीक है। अब RF1 release हो गया, but still it is bound to your RF3 and RF3 will what happen? RF3 when it is bound, see RF3 when it is bound to GDP, okay, जब GDP के साथ bound है, it has got high affinity for your RF1 and the ribosome, okay. अब क्या हुआ है, exchange had happened, so GDP replace होके GTP आ गया है, okay. Now this GTP will undergo hydrolysis, correct. This hydrolysis will result in the release of your RF3 also, and this will have no further affinity for your ribosome mRNA and the tRNAs. So this is how your translation is getting terminated. Clear? यहाँ पे कोई doubt? इतना clear है, like how translation is getting terminated. So this is how it is happening in the case of the prokaryotes. Should I repeat or is it clear to you, Tanvi?
Yeah, ma'am, clear.
It's clear, na? Good. Okay. Now we need to talk about how to recycle the ribosomes, tRNA and the mRNA present. ठीक है? एक बार when the cell understands that the mRNA has completed its fate, यानी कि उतना protein expression हो गया है, then the mRNA should undergo degradation. ठीक है? But it doesn't mean that every time the tRNA and the ribosomes are also getting degraded. Okay, so cell kya karega? The information has been passed, it has got converted into the polypeptide chain. Further, it will get properly folded into a protein, and the protein is going to do its activity. ठीक है? ये तो हो गया आगे का सिनेरियो. पीछे क्या हो रहा है? We don't make ribosome and tRNA every time because it is waste of energy. When you can recycle it, why to synthesize or generate more copies of ribosome and tRNA again and again? Because we can use it same way, right? हम हमेशा उसको use कर सकते हैं. So we need to recycle these. Now in order to recycle all, okay, in order to recycle all, if you remember in the previous slide we saw that RF1 को रिलीज करने के लिए हमारे पास RF3 था, ठीक है. RF3 when GTP hydrolysis happened, RF3 also got released. अभी क्या left over है वहां पे? आपका large subunit, okay, and small subunit, and the mRNA is present, if you remember. So now we need to recycle this. mRNA का future क्या है? That it needs to get degraded. Okay, because the protein or the information has been passed, but ribosome can get recycled, right, for the another round of translation. So we need to remove, or we need to detach or dissociate your large subunit and the small subunit. अब इसको करने के लिए, like for the recycling of the ribosome, we have got another factor which is known as ribosome recycling factor. Okay, it is called RRF. Okay, now after the release of the polypeptide chain and the release factor, the ribosome is still bound to the mRNA and two deacylated tRNAs, right? आपका एक P में था और एक E में था, if you remember. अब elongation के time में what we have discussed is that whenever EFG comes and undergoes a GTP hydrolysis, there is a opening of the EFG structure, जिसके वजह से क्या होगा? पहले तो structure आपका ऐसे होता है. Okay, when GTP hydrolysis happen, it becomes like this. So it pushes from A to P, right? And P का आपका E में चला जाएगा. So that's how elongation was happening. Now here also, in order to release your tRNA, which is present on the E side and the P side, we need the activity of your EFG, because this is meant for translocation. ठीक है? इसका काम क्या है? Translocate करना. So same way here also we need to free the tRNA. So we again need the activity of EFG. Okay. So here in prokaryotes, a factor known as ribosome recycling factor, which cooperates with EFG and IF3 to recycle ribosomes after the polypeptide release. Now, then, do you remember what was the function of IF3, initiation factor 3? Can you recollect what was the function of IF3? I know थोड़ा gap हो गया है, but still, are you able to recollect or not?
No, ma'am.
Okay, no problem, no problem. It's okay because थोड़ा सा time हो गया है ना. It's okay. So I'll tell you. So EFG का काम just now I have discussed that it is meant for the translocation, right? So when we say translocation with respect to translation, देखो, in future also we are going to use this term translocation a lot, especially in the case of the animal physiology and plant physiology. So there it has got different meaning. Now here with respect to mole bio translation, translocation means it is shifting of the tRNA from P to E and E to outside. Okay, sorry, A to P and P to E. Okay, so this is the meaning of translocation, which is done by elongation factor G. Now, IF3, if you remember, it was the to prevent the immature binding of the large subunit to your small subunit. Okay, there was one figure in that you can see that small subunit आपका ऐसा रहता है, यहाँ पे आपका IF3 आके bind करता है. ठीक है, now because of the presence of the IF3, large subunit was not able to bind. Okay, so that was in order to prevent the immature or the before time binding of the large subunit to your small subunit. Okay, अब हमें IF3 की क्यों जरूरत है? Because we are going to dissociate the large subunit and the small subunit. अब वापस वो interact ना करे, इसलिए IF3 will go and bind over there. ठीक है, so now we need the requirement of IF3. EFG का काम है to make tRNA free from the ribosome complex and IF3 to prevent the binding or the interacting of large subunit back to the small subunit. ठीक है, okay. So RRF, that is ribosome recycling factor, it goes and binds to the empty A site of the ribosome where it is going to mimic like a tRNA. So this is going to recruit your EFG GTP to the ribosome, and in that event, it mimics EFG function during the elongation. So when it is going to stimulate the release of the unchecked tRNA bound on the P and E side. ठीक है, so कैसे, if you remember here also जब हम elongation पढ़ रहे थे, so the tRNA amino acylated tRNA is present here and the peptide tRNA is present. Now we need to shift this polypeptide chain from P side to A side has been shifted, correct? Now A side का जो आपका tRNA है that should go to P side and P का जो है वो आपका E में जाना चाहिए. ठीक है, तो ये काम करने के लिए EFG यहाँ पे आके bind करता है. It upon GTP hydrolysis it gets open up like this. तो ये इसके ओपनिंग की वजह से tRNA जो A साइड में वो आपका P में जाता है, P में जो है वो A में जाता है. तो same thing is gonna happen here also. Okay, because here the RRF is going to act as a tRNA. Now, once the tRNAs are removed, there is a EFG GDP because hydrolysis ho gaya and RRF. They will get released from the ribosome along with the mRNA. So at the end, what had happened? आपका mRNA free हो गया, small subunit free हो गया, large subunit free हो गया, and tRNAs are also now free. Correct. So now this IF3 initiation factor 3 will come and it is going to bind to the smaller subunit. Okay, and it will make sure that the large subunit, the free large subunit does not come back and bind. अब ये कब bind करेगा? जब आप second round of translation is going to start. ठीक है, then again you're going to recruit all the initiation factors like 1, 2 and all these will come and start making the pre-initiation complex. Then only IF3 will get released and last subunit will come. Is this much clear, Tanvi? It's a like long procedure. So what I suggest is like you hear the recording एक साथ, ठीक है? Just watch it continuously so that you get the entire idea of translation. Okay. So the concept is clear, I believe, even if you don't remember the name of the factors, but I hope the concepts are clear. Yes?
Yes, ma'am.
Okay. So this is the same thing now with the help of the diagram. So what do we see here? That your ribosome releasing factor, if you see, it has got the structure similar to your tRNA. It goes and binds to the A site. ठीक है, यहाँ पे आपका EFG GTP आ रहा है. So you can see, जैसे GTP hydrolysis होगा, this portion of the EFG, okay, ये जो portion है ये आपका open up हो जाएगा. So because of which it is going to push the tRNA. Okay. So you will see when the moment it gets pushed, what will happen? This is going to remove your free tRNAs. और जब ये भी release हो जाएंगे, IF3 will come and bind to your smaller subunit. Hence, mRNA is free now. All your release factors are free, and the large subunit. So they are ready for now second round of translation. Okay. Now, Do you want to ask any questions before I proceed? Okay, so let's compare now. So when I say let's compare means हम लोग भी compare करने जा रहे हैं कि कौन कौन से factors आपके eukaryotes में present है, prokaryotes में present है, ठीक है? And this table are really very important because आपके वो जो part B में जो questions आते हैं 2-2 marks के वो इसी पे base आते हैं, ठीक है? Rest all they will ask what will happen if this particular antibiotic has been used. So there will be another table at the end of the mole bio session where I'll provide you like what are the antibiotics required in the prokaryotes and eukaryotes to inhibit the activity of the DNA replication, transcription and translation. Okay. Okay. So now initiation factors, IF1 है यहाँ पे, so you can see that is a small letter E for the eukaryote. Then again it is same IF1 and A is a which is blocking the A site. ठीक है? IF2 again over here. It is going to help in binding the methylated tRNA. Then IF3. So it is going to have all the subsequents over here are there. First to bind and prepare 40S for the subsequent factors. And in the case of here, it is going to help in the dissociation of these IF2 and the EIF3. So here the function is also given. Now, यहाँ पे क्या होगा यूकैरियोट्स के केस में you have got the additional initiation factors are there. So if you remember, these ones are required to prepare your ribosomes and these ones are required to prepare your mRNA. So जब ये दोनों साथ में आते हैं तब आपका यूकैरियोटिक pre-initiation complex बनता है which is having the 43S का subunit. Usually it is 40, right? So when all the mRNA along with these elongation initiation factor comes together and binds to the ribosome, it forms a pre-initiation complex of 43S. Okay. Now coming to the elongation factors. So here you have got the EF2, TS and G. So 2 का काम क्या होता है? To bring the aminoacylated tRNA to the ribosome. This helps in the exchange of the GTP and G का for the translocation. So similarly, we have got over here the homologs in the case of eukaryotes also. So eukaryotes में भी है आपका eukaryotic elongation factor 1 alpha, here 1 beta gamma, and here eukaryotic elongation factor 2. ठीक है? So simple to remember. Last, so prokaryotes में all the three are there, so they have got different. So there was another table in which I have shown like RF1 किस codon को recognize करता है, RF2 कौन से stop codon को recognize करता है, ठीक है? Now in the case of the eukaryotes, it is the only single release factor which recognizes all the codons and उनका function क्या है? Release of the polypeptide chain or in short termination of the translation. Okay? Yeah. So that's all, Tanvi. This was the that was all about the translation, the termination. Any other doubts are there? Would you like to ask, or we can stop here because translation is over here.